Ja, einen wunderschönen Nachmittag von meiner Seite. Danke Ihnen allen, dass Sie zu dieser ÖGMBT Award Verleihung gekommen sind. Wenn Sie möchten, es ist mir zwar gesagt worden, die Preisverleihung soll in Deutsch stattfinden, aber ich kann es gerne auch in Englisch machen. Also vielleicht hätten wir jetzt noch Gelegenheit für einen Switch, aber sonst bleibe ich bei Deutsch wie vereinbart. Mein Name ist Viktoria Weber, ich bin Vizepräsidentin der ÖGMBT und ich darf heute, es ist mir eine große Freude, hier in Präsenz, trotzdem in dieser speziellen Situation, unsere ÖGMBT Awards vergeben zu dürfen. Ich freue mich, dass ich Sie alle hier in dieser, wenn auch kleinen Gruppe, aber doch persönlich begrüßen darf und ich möchte mit unseren Gästen vom Ministerium und mit unseren Unterstützern beginnen. Ich begrüße Herrn Sektionschef Florian Frauscher und Frau Abteilungsleiterin Dr. Ulrike Unterer. Herzlich willkommen und vor allem auch herzlichen Dank. Es wird davon noch die Rede sein, auch für die Unterstützung, die Sie uns bei diesen Preisen immer angedeihen lassen. Ich glaube, das ist eine sehr, sehr wichtige Unterstützung für uns, unsere Mitglieder, für die gesamte Gesellschaft und ich glaube, es ist auch wirklich ein wichtiges Zeichen für die Bedeutung einer solchen wissenschaftlichen, doch großen Fachgesellschaft im Life Science Bereich. Ich darf natürlich auch alle Preisträgerinnen und Preisträger sehr herzlich begrüßen. Wir hatten 48 Einreichungen, 5 konnten wir auswählen und 5 dürfen wir heute auszeichnen. Das freut uns sehr. Ich glaube, jeder, der viele Jahre und Monate und ja, lange Zeit im Labor verbringt und sich in ein Thema so vertieft, freut sich, einmal hier stehen zu dürfen und für diese Arbeit auch wirklich einen Preis entgegennehmen zu dürfen und diese Auszeichnung entgegennehmen zu dürfen, die der Arbeit dann auch neben dem Interesse, mit dem man sie verfolgt, einfach auch die notwendige Sichtbarkeit gibt. Und ich glaube, auch das ist ein wichtiges Zeichen, das wir hier setzen können, eben Sie auszuzeichnen für Ihre Arbeit. Die Preisträgerinnen habe ich schon begrüßt. Ich möchte auch all unseren Sponsoren ein Dankeschön sagen. Wir werden sie dann bei den einzelnen Preisen auch noch bei der Preisverleihung dabei haben. Und es ist mir auch ein Anliegen, meinen Kolleginnen und Kollegen von der ÖGMBT-Geschäftsstelle sehr herzlich zu danken. Frau Kassedow ist heute nicht bei uns, kann nicht bei uns sein, aber ich bin sicher, sie verfolgt mit großer Spannung die Veranstaltung jetzt aus dem Off. Aber Frau Bauer und Herr Glaser, herzlichen Dank für die ganze Arbeit, die Sie sich hier gemacht haben und die dazu beiträgt, dass wir heute diese Veranstaltung so durchführen können. Ja, es war eine schwierige Entscheidung für uns im Frühjahr, als wir beisammengesessen sind im Lockdown, uns via Zoom getroffen haben und überlegt haben, der Kongress, der kommt in die Nähe, was machen wir? Damals die Entscheidung zu treffen, wir sind lieber auf der sicheren Seite und werden von vornherein ein Format planen, von dem wir wissen, dass wir es dann auch umsetzen können und wir sind jetzt sehr froh darüber, dass wir so geplant haben. So ist es heuer ein besonderer Kongress, weil er zum Großteil online stattfinden wird und muss. Wir haben aber aus der Not eine Tugend gemacht. Es gibt eine 3-tägige Kick-Off-Veranstaltung, die mit heutigem Tag begonnen hat. Also heute ist der erste Tag, es gibt morgen und übermorgen dann den 2. und 3. Tag der Kick-Off-Veranstaltung. Und wir haben ein neues Format kreiert, das sind die sogenannten Life Science Tuesdays. Wir haben uns überlegt, eine virtuelle Veranstaltung kann nie denselben Interaktionscharakter haben wie eine persönliche, eine Veranstaltung mit persönlicher Präsenz. Ich weiß noch gut, Frau Unterer, wie wir voriges Jahr in Salzburg gestanden sind und die Atmosphäre ist einfach eine andere. Aber wir haben gesagt, gerade für unsere jungen Kolleginnen und Kollegen und für unsere vielen jungen Mitglieder, die wir in der Gesellschaft haben und die wir ja auch fördern möchten, wäre so ein Format, das sich über einen längeren Zeitraum hinzieht, vielleicht eines, von dem Sie sehr viel profitieren können. Und diese Life Science Tuesdays werden von Oktober 2020 bis März 2021 jeden Dienstagnachmittag stattfinden zu einem bestimmten Thema. Und es wird möglich sein, dort online dabei zu sein, online Vorträge einzureichen und eben eigentlich auf einem sehr leicht zu erreichenden, auf einer sehr leicht zu erreichenden Ebene jeden Dienstagnachmittag einmal im Monat eben diesen Life Science Tuesday mit uns zu machen zu spannenden Themen. Sie haben sie hier alle im Programm. Das ersetzt dann quasi die Sessions, die wir sonst bei der ÖGMBT beim Kongress eben an einem Nachmittag oder an mehreren Nachmittagen hätten. Ja, ich darf mich auch noch bedanken dafür, dass wir hier sein dürfen bei Frau Bea Cohen-Kriesmer vom Department für Applied Life Sciences, dass sie uns die Räumlichkeiten hier zur Verfügung stellt und dass wir uns hier wirklich, danke, dass wir uns hier wie zu Hause fühlen dürfen. Vielen Dank. Ja, ich habe schon ein bisschen erzählt über das neue Format, über das Kickoff-Meeting jetzt, über die 3 Tage Kickoff, die wir noch haben, über die insgesamt 12 Life Science Tuesdays. Und möchte vielleicht noch ein paar Worte in aller Kürze darüber sagen, warum machen wir das überhaupt, warum vergeben wir überhaupt Preise. Wir vergeben Preise, weil es eines unserer wichtigsten Ziele ist, als größte Life Science Gesellschaft Österreichs, den wissenschaftlichen Nachwuchs zu fördern. Und das kann man tun, indem man einerseits Rahmenbedingungen schafft, in denen Wissenschaftlerinnen und Wissenschaftler arbeiten können. Aber dazu gehört auch mehr als das, es gehört eben auch eine gewisse Sichtbarkeit dazu. Und diese Sichtbarkeit möchten wir unter anderem eben mit diesen ÖGMBT Awards geben. Und wir freuen uns hier auch wirklich noch einmal ausdrücklich über die Unterstützung des Ministeriums für Digitalisierung und Wirtschaftsstandort, dass Sie uns hier auch mit diesen Preisen so unterstützen. Ich glaube, es ist ein Zeichen auch darüber hinaus, nämlich es geht auch ein bisschen um den Stellenwert, den die Wissenschaft in unserer Gesellschaft im Allgemeinen hat. Gerade in Zeiten wie diesen, gerade in einer so krisenhaften Situation, wie wir sie jetzt in den letzten Monaten erleben mussten, glaube ich, ist sehr klar geworden, was die Rolle der Wissenschaft ist. Das ist bei weitem nicht der Elfenbeinturm, das sind nicht die Universitäten, wo sich die Wissenschaftler mit irgendwelchen Fragen beschäftigen, sondern da geht es wirklich darum, ganz praktische Antworten zu finden. Es geht manchmal auch darum, das ist dann auf der einen Seite immer verständlich, auf der anderen Seite muss man dann immer bremsen, ganz, ganz schnell Lösungen anzubieten. Also da muss man dann auch, und das hat mir auch sehr gut gefallen, wir hatten letztens die Freude, bei der 25-Jahr-Feier der Christian-Doppler-Gesellschaft auch über dieses Thema zu sprechen, dass dann auch, glaube ich, auch die Geduld gefragt ist und nicht alles sofort abrufbar ist. Aber ich denke, es ist ein wichtiges Zeichen, das wir hier setzen für die Wissenschaft und für den Stellenwert, den Wissenschaft in unserer Gesellschaft hat. Und gerade auch aus diesem Grund haben wir zu den Preisen, zu den vorher 4 Preisen, die wir hatten, seit 3 Jahren nun einen 3. Preis dazugefügt. Und zwar einen Preis, der speziell darauf aufmerksam machen soll, was die gesellschaftliche Wirksamkeit von Forschungen betrifft. Also diesen Preis für Societal Impact, mit dem wir diese Facette auch noch einmal besonders betonen möchten. Ich danke auch allen Jurorinnen und Juroren. Ich habe schon gesagt, wir hatten insgesamt 48 Einreichungen und von diesen 48 Einreichungen haben wir 5 auswählen müssen, was nicht einfach war. 16 Jurorinnen und Juroren haben uns im Corona-Sommer dabei unterstützt. Und von den 5 Einreichungen, das ist vielleicht auch noch interessant, freuen wir uns, dass wir mehr als die Hälfte, also 5 ist eine ungerade Zahl, daher kann es nur entweder weniger oder mehr als die Hälfte sein, aber dass wir unter den Frauen 3 Frauen als Preisträgerinnen haben. Also hier sehen wir nochmal das Format oder die verschiedenen Kategorien, die wir haben. Sie wissen das. Es gibt den Research Award 3x 3.000 € und den PhD Award mit 2x 1.000 €. Und von jedem der beiden Preise gibt es jeweils eine Kategorie angewandte und Basic, wobei das gar nicht immer weder für die Einreicher noch für die Juroren ganz einfach zu trennen war, weil auch hier sieht man, also es gibt überall eine Anwendung drinnen und es gibt überall die Grundlagenaspekte dazu, aber bei manchen war es eindeutig und so haben wir uns dann am Schluss dann doch nicht so schwer getan, die Preise auch wirklich richtig zuzuordnen und schon hervorgehoben habe ich den Special Award für Societal Impact, den wir auch bereits zum 3. Mal vergeben. Ich darf jetzt ja, hier sind die Preise nochmal aufgelistet und ich darf jetzt die Grußbotschaft von Frau Ministerin Margarete Schramböck hier in den Saal bringen und in die Öffentlichkeit bringen, bevor ich Sie dann um Ihre Grußworte bitte. Und ich würde das Video hier starten für die Grußbotschaft.
Sehr geehrte Frau Vizepräsidentin, liebe Preisträgerinnen und Preisträger, sehr geehrte Damen und Herren. Die jetzige Corona-Krise hat uns gezeigt, wie wichtig Wissenschaft sowohl in der Grundlagenforschung wie auch in der angewandten Forschung für Österreich, Europa und die Welt ist. Besonders der Life Science Bereich spielt hier eine großartige Rolle. Es geht darum, Impfstoffe zu entwickeln, es geht darum, Medikamente zu entwickeln, es geht darum, die Möglichkeiten der heutigen Erkenntnisse zu nutzen. Sie sind besonders talentiert darin. Sie sind die Vorreiter, die Vordenker, die Wissenschaftlerinnen und Wissenschaftler, die Probleme lösen werden, die wir vielleicht heute noch nicht lösen können oder vielleicht noch gar nicht kennen. Der Life Science Award spielt für mich eine ganz wesentliche Rolle. Wir wollen Ihr Talent auszeichnen, Sie als Nachwuchs ehren und Sie auch fördern für die Zukunft Österreichs und der Menschen. Ich gratuliere allen, die heute ausgezeichnet werden. Ich bin stolz auf Sie und ich wünsche Ihnen alles Gute für Ihre Zukunft und für Ihre Forschung.
Dann darf ich bitte Sie, Herr Frauscher, herausbitten um Ihre Grußworte. Haben Sie ein Mikrofon?
Ich höre das Mikrofon gerade noch. Zuschauer daheim auch. Sehr geehrte Damen und Herren, hier am Wiener Biocenter, sehr geehrte Damen und Herren zu Hause. Es freut mich sehr, dass ich heute auch physisch hier sein darf bei dieser ersten virtuellen Jahreskonferenz der ÖGMBT. Es sind besondere Umstände, in denen die diesjährige Tagung abläuft, aber trotzdem sehr innovativ, wie ich finde. Auch Gratulation dazu, dass das so toll funktioniert in der Planung, dass das Ganze trotzdem durchgeführt werden kann. Wir wissen von vielen anderen Konferenzen, die ganz abgesagt wurden, und so ist es doch, glaube ich, ein tolles und ein wichtiges Zeichen, dass diese Konferenz auch stattfindet. Ich finde auch sehr spannend, Frau Weber, diese Life Science Tuesdays, die Sie vorgestellt haben, weil das dazu führt, dass es nicht nur ein einmaliges Event ist, das jetzt nur an 1 oder 2 Tagen stattfindet, sondern es sich auch über mehrere Monate, ich glaube bis März nächsten Jahres, dann zieht. Es ist oft auch viel leichter, gerade wenn es digital stattfindet, wenn man nicht 2 Tage durchgehend an einem Bildschirm ist, sondern es in kleine Blöcke auch teilt und so auch länger noch einen Austausch garantieren kann. Wir als Wirtschaftsministerium fördern seit 2017 schon diesen Preis. Wir stehen für standortrelevante Forschung. Was verstehen wir unter standortrelevante Forschung? Da gehört sowohl die angewandte dazu, da gehört die Grundlagenforschung dazu, aber alles, wo Unternehmen, wo Wirtschaft und Forscherinnen und Forscher kooperieren, überall, wo Produkte entstehen, Forschung, Ergebnisse entstehen, die dann später genutzt werden können, um diese in Therapien überzuführen, in Produkte überzuführen. Das ist unsere Aufgabe, das zu unterstützen und das zu fördern. Und das machen wir sehr gerne, und zwar immer in einem Bereich, wo es um Exzellenz geht. Das ist uns auch ganz wichtig. Wir wollen sozusagen nicht mit der Gießkanne fördern, sondern wir wollen Exzellenz in Österreich fördern. Und das funktioniert in verschiedenen Programmen, bei verschiedenen Gesellschaften. Sie kennen sicher unsere Komet-Zentren, bei denen wir das machen. Sie haben schon die Christen-Doppler-Gesellschaft erwähnt, bei der das passiert. Wir haben eine jüngere Initiative, Wings for Innovation, wo wir mittels eines Fonds auch Ergebnisse aus der Grundlagenforschung im Life Science Bereich überführen wollen, unterstützen wollen in Therapien, in kommerzielle Produkte. Es gibt eine sehr breite Palette an Unterstützungsmöglichkeiten, aber am Ende geht es immer um die standortrelevante Forschung und um Exzellenz. Dazu zählt auch die Anerkennung von wissenschaftlichen Leistungen durch Preise. Es gehört dazu das Sichtbarmachen. Sie haben es auch schon genannt, wie wichtig es ist, junge Talente vor den Vorhang zu holen und auch in die Breite zu gehen mit dem, was wir tun. Ein bisschen nach dem Motto, tue Gutes und sprich darüber. Wir haben ein bisschen in Österreich das Thema, obwohl wir eigentlich in F&E sehr gut sind, dass das Thema noch immer ein bisschen zu wenig in der Öffentlichkeit steht, noch immer ein bisschen zu wenig Aufmerksamkeit geschenkt wird, obwohl es so enorm wichtig ist. Nicht nur jetzt in Zeiten einer Corona-Krise, einer Pandemie, wo doch die Wissenschaft und die Forschung wieder mehr ins Zentrum der Aufmerksamkeit gerückt ist. Aber grundsätzlich ist es wichtig, einfach noch mehr über Forschung, Entwicklung zu kommunizieren, zu sprechen. 2/3 unseres Wirtschaftswachstums und damit unseres Wohlstands kommen von Forschung und Entwicklung. Und damit ist dann auch jeder Euro, den wir investieren in Forschung, ein gut investierter Euro. Und daher auch die Bitte an Sie, kommunizieren Sie, gehen Sie auch nach außen, teilen Sie Ihre Ergebnisse, diesen Preis auf Social Media, kommunizieren Sie drüber, denn es ist wichtig, auch diese Dinge, die Sie hier machen, nach außen zu tragen. Ganz besonders, glaube ich, ist es wichtig, von den heutigen Preisträgerinnen und Preisträgern zu erzählen. Es freut mich auch sehr, dass mehr als die Hälfte weiblich ist. Auch das ist uns im Wirtschaftsministerium, der Margarete, die Sie gerade gesehen haben, ein ganz besonderes Anliegen. Das heißt auch hier ein ganz wichtiges Signal an die Forschungslandschaft, die Forscherinnen und Forscher mit diesen Preisen auszuzeichnen. Ich darf mich bedanken noch bei der ÖGMBT für die Organisation der Konferenz, für die Organisation der Preise und darf mich bei der Jury auch bedanken, wo sicher auch viele zusehen und bei allen tatkräftigen Helfern, die beigetragen haben zum Erfolg der heutigen Konferenz. Und mein letzter Satz gilt den Preisträgern. Herzlichen Glückwunsch, alles Gute. Dankeschön. Entschuldigung. Wir müssen wieder tauschen, sonst hört man sie nicht.
Wir kommen jetzt zum eigentlichen Sinn und Zweck dieses Nachmittages, nämlich wirklich zur Verleihung der Preise. Und wir freuen uns auch, dass jeder der Preisträger, die Preisträgerinnen uns auch kurz über seine Arbeit berichten wird, also in Form eines Vortrages zu den einzelnen Preisen. Wir werden zuerst beginnen mit den Research Awards und werden zunächst diese 3 Awards verleihen. Dann haben wir die 3 Vorträge, die 3 Vorstellungen der Gewinnerprojekte, bevor wir dann zur Verleihung der PhD Awards kommen. Und ich darf herausbitten die Gewinnerin Research Award Basic Science, Laura Gallego. Und darf auch Sie, Herr Sektionschef, noch einmal herausbitten, dass wir gemeinsam diesen Award übergeben können. Bitte. Wir dürfen ihn leider nicht von Hand zu Hand übergeben, aber herzlichen Glückwunsch, greifen Sie zu, er gehört Ihnen. So, dann gehen wir zur zweiten. Okay, dann können wir das leider, der Beamer hat gerade ein kleines Problem, das heißt, wir müssen das jetzt ohne Begleitvideo machen, was aber vielleicht für die, die vor dem Beamer stehen, besser ist. Dann darf ich unsere zweite Gewinnerin herausbitten, Research Award Applied Research, Christina Mayor-Ruiz. Dankeschön. Herzlichen Glückwunsch. Wir freuen uns auf den Vortrag danach. So, dann kommen wir zum Special Award, Excellence in Societal Impact. Also da ging es um die gesellschaftliche Wirksamkeit im Besonderen. Herr Thomas Gassler. Und was ich Ihnen jetzt allen noch schuldig geblieben sind, waren die Titel dieser oder die Themen. Aber nachdem Sie uns gleich jeweils in Ihren Vorträgen darüber erzählen wollen, bitte um Nachsicht, dass ich das jetzt nicht extra noch erwähnt und vorgelesen habe, aber wir freuen uns auch dann auf Ihren Vortrag. Gratuliere. Gratuliere, herzlichen Glückwunsch. Ein Foto brauchen wir noch. So. Und das heißt, wir kommen jetzt zu den Vorträgen und ich darf Frau Gallego bitten um die Vorstellung ihres Projektes. Der Beamer funktioniert wieder und so, jetzt darf ich das trotzdem noch vorstellen. Also das Gewinnerprojekt Phase Separation Directs Ubiquitination of Gene-Body Nucleosomes und bitte um den Vortrag. Ja, ja.
So, good afternoon, everyone. I would like to start today by saying thank you to all the organizers and the people that are involved in making this ceremony to happen in the conditions that we currently have. During the last years, I focused in studying the mechanism of chromatin ubiquitination and concretely on histone H2B ubiquitination. This is a very concrete modification that targets the residue lysine 123 on H2B. And is mediated by two enzymes, FRAT6, the E2 conjugating enzyme, and BRI1, the specific E3 ligase. This modification is not a signal for protein degradation, which is the common pathway for ubiquitinated proteins, but is rather recognized by other chromatin modifiers that would be recruited and therefore exert their functions, such as methyltransferases. So together, methylation and ubiquitination can control chromatin compaction and therefore gene expression. In the last years, I published a model also studying how the specificity towards the lysine 123 residue was achieved. And we showed that it's the recognition of the E3 ligase towards the substrate, the nucleosome, which brings the E2 Rad6 in such an orientation that the lysine 123 is the closest residue to get targeted. But this model doesn't explain how in the complex context with chromatin in vivo this specification is achieved. And moreover, it doesn't explain how this modification is related to transcription. Therefore, there must be some other regulators in vivo, complexes and protein, that might function in this regulation of this process. And therefore, we've started to work with large one, because this protein was described to be also essential for chromatin ubiquitination in yeast. Large one is a rather small protein that is only 300 amino acids, and in the C-terminal it has a predicted coil-coil. But up to 70% of the protein is disordered, so we call it the IDR, for intrinsically disordered region. This coil-coil domain we show by different assays that is mediating a direct interaction with the E3 ligase, BRI1, as you can see here in this binding assay, where we have tagged different constructs of large one, and we use it as a bait for the interaction with the E3 ligase. So what's the rest of the protein doing, these 70% residues? So we optimize the purification of the protein, and when we check under the microscope, this is what was happening. So the protein, the IDR is mediating phase separation. And this process is something that we see every day when we are dressing our salad with olive oil and vinegar. But in the protein field, it means that your sample will have two different phases. One that is enriched in protein, because the protein-protein interactions are energetically favorable versus the protein-water interaction. And these are the ones, the droplets that you can see, surrounded by the buffer that is depleted of protein. So the IDR is mediating the phase separation, as you can see here in these micrographs. Phase separation is currently involved in many different processes in the cell, and there are more and more proteins that are known to mediate this process, creating membrane-less condensates in the cytoplasm and also in the nucleus of the cell. But the big question that remains in the field is what's the function of these proteins and why is the phase separation important, if so. So now we have a protein that is essential for chromatin ubiquitination and is mediating phase separation. So we wanted to address whether actually the phase separation was functionally important for large one. And we addressed this question initially by studying the interaction of BRI1 and large one under phase separating conditions. So here we have BRI1 in green with the GFP, and you can see that they both interact and BRI1 form a shell with the thickness that is increasing proportionally to the concentration of the protein that is added. And this shell that is formed around the large one condensate is specific for the interaction with the coil-coil, because when we have the same experiment done just with the IDR, we see that BRI1 is partitioning inside the condensates. But BRI1 is only one of the components. What is happening with the other components of the ubiquitination reaction? So we checked the behavior and the partitioning of the Rad6 enzyme, and we saw that this first recruited to the BRI1 shell by a direct interaction with BRI1 and slowly diffuses into the large one core. And then with chromatin, we reconstituted chromatin in the lab, and we could also see that initially there is a direct interaction with the shell, and again is slowly diffusing into the core. So now we have a condition where all the components of the ubiquitination reaction are concentrated in the condensates. And therefore, we ask whether this was having a function in the reaction itself. We performed in vitro ubiquitination reactions, and we could see that when we were adding phase separating large one, there was an enhancement of ubiquitination levels. And this is very clear from the results and also from the quantification. And when we were doing the same experiment, just adding a construct that is still able to interact with the A3 ligase, but not phase separating, this enhancement was not observed. So the phase separation is important in our case for this process. And this is all very striking, but it is all in vitro. So then we decided to ask whether these condensates are also present in vivo, in cells. And for that, we performed the bimolecular fluorescent complementation assay. And in this assay, we split the Venus tag, here in yellow, in two parts. One fused to the coil-coil of large one, and the other one to BRI1. Only when these two proteins will interact in yeast, we will have a fluorescent signal. And in our case, when we use the large one full length, able to phase separate, we could observe the formation of the bright punta. In contrast to the construct that is still interacting with BRI1, where the fluorescence signal was very homogeneous, suggesting that these condensates are actually formed in vivo. So then the next question was to ask, where are they recruited to chromatin? There are many different techniques that you can use to study how the protein is recruited to chromatin and where by chromatin immunoprecipitation assays. And here I just show an example of a nucleosome profile made by the MNA-seq, just to say that along the genome, the nucleosomes in all the genes have a very specific positioning. Here we have the nucleosomes in the promoter, followed by the nucleosome-free region, where all the components of the initiation of transcription will assemble. And after the transcription start site, we have the nucleosomes in the gene body, all well positioned. In our case, we decided to use ChIP-EXO because this technique gives a really high resolution to the base pair level of where your protein of interest is recruited. And here you can see the profile that we obtained for the global levels of H2B ubiquitination in wild type. And also we can see that there are 2 peaks for each nucleosome because the resolution is such that you get even the different positioning of the 2 histones that are present in the nucleosome. So as a result, we saw that the ubiquitination is enriched in the gene body of the nucleosome. And then we studied how the different constructs of large 1 would look like. And we observed that the deletion of large 1 affects dramatically the ubiquitination levels in yeast. And the construct that is not able to phase separate is affected also in the ubiquitination in the levels of the gene body, further of the +1 nucleosome. So with all these results together, we propose a model that would be the current model for chromatin ubiquitination, where the large 1 protein and BRI1 form layer condensates, where we have a disorder core and a catalytically active shell that is able to recruit the substrate, chromatin, and all the components of the ubiquitination machinery for optimal levels of ubiquitination in vivo. And with this, I would like to thank my group, the Kohler group, and especially the people that was directly involved in this project, our collaborators in the US and here in campus, and all of you for your attention. Yeah.
Thank you very much for the great presentation. It was very interesting, and yeah, thank you also for sticking to your time, which is the more important when we have an online meeting, but thank you and congratulations again. Ja, dann darf ich die nächste Vortragende herausbitten. Genau, Christina Major-Ruiz vom CEM, und ich gebe das Mikrofon gerne weiter.
Okay, so good morning, everyone. And first of all, I want to say that I'm extremely grateful to the ÖGMBT for this award. I feel really honored, and also thank you very much for the incredible support to young scientists every year. And with that, I would like to present the strategy that we established that for the first time allows the rational discovery of a new kind of drugs with really an incredible therapeutic potential that are called molecular glues. And to contextualize a bit this story, I would like to introduce to you the concept of targeted protein degradation. So this is a strategy that is based on the use of drugs that we call degraders that induce proximity between an E3 ubiquitin ligase and a target protein of interest. And as a result, this target protein is ubiquitinated and then degraded by the proteasome. And this shift from inhibition to just binding has allowed the targeting of proteins that were considered undruggable. And something important is that most of the degraders available so far, they recruit E3s of this family, the calin-rel ligase family, which, and these ligases are modular complexes as depicted here. And please keep in mind that this component, the so-called SASTRO receptor, is the part of the E3 ligase that is recruited by those degraders. So if we talk about degraders, there are 2 different kinds. So we have PROTACs that are modular molecules, and then we also have molecular glues. And these molecular glues are non-chimeric small molecules, so there is not really a rational design. And what they do is that they induce highly cooperative de novo protein-protein interactions. And they have proof that it's possible to degrade undruggable proteins. And the best example are thalidomide and its analogs called IMiDs that now are known to induce degradation of otherwise undruggable transcription factors. And the thing is that the few molecular glues available so far, the discovery of them was driven by serendipity. So there were not really rational strategies behind those discoveries. And we were convinced that one key step for the field will be to come with the strategies to, in a rational way, identify those molecular glues. And that's exactly what we tried to do with this project. So we wanted to come with a strategy applicable to many trees at the same time, target agnostic, cell-based, and as scalable as possible. And to guide you through the rationale that we followed to come with such a strategy, I want to briefly mention this work that we published last year, where we set out to identify the global genetic determinants governing targeted protein degradation. And for that, we took 5 degraders, including molecular glues and products, and we did CRISPR resistance screens. And when we contextualized our hits in this beautiful network, we could segregate SER from drug-unique genetic determinants. But what is key for my talk today is that we did find one case, UB2M deficiency, that confers resistance to all the degraders tested. And what is this UB2M actually doing? So kalium religases are only active if they are nedylated. So this is a post-translational modification that is dependent on an enzymatic cascade. And UB2M is essential in this cascade. So when we generated UB2M deficient models in KBN7 cells, so a leukemic cell line, we realized that more than 200 kalium religases were inactivated. And why is this important? So, well, I just mentioned that UB2M deficiency confers resistance to known degraders like this product. So we thought that we could use that differential viability to actually look for potential novel molecular glues. And this is the design of the screen that we did. So we took from the same library, which is drug library, which is called placebo, we took all the compounds that were cytotoxic or cytostatic, and we tested them at 2 doses in both wild-type cells and UB2M deficient cells. And this is the summary of the screen. So we did find several molecules that were affecting viability of wild-type cells, but not viability of these UB2M deficient cells. So all of those here were potential molecular glues. And we tested that this differential effect was also true in a dose-resolved manner, and not only that, also in an additional hyponedylated model. So this time is colon cancer cells. So this gave us confidence to try to profile these molecules, these potential molecular glues. So starting with the same one, if this is really a molecular glue, what we are facing here is a dual target identification. So on the one hand, we have to identify the potential kalium religase involved, and for this, what we did was CRISPR-resistant screens. So we team up with the Sugar Lab to design a kalium religase-focused S-guide RNA library. And then in order to try to identify the potential targets, so the protein degraded, what we did was expression proteomics. So what we learned with the CRISPR screens was that we found, again, guides against UB2M, so there was a nice positive control, and we could find that this kalium religase, the GAT15, was key for the mechanism of action. And what about the target? So when we did expression proteomics, we could observe that the same one was inducing a very selective destabilization of this essential splicing factor, RBN39. And this mechanism of action actually had been already described. So this one molecular glue called indisulam that is structurally similar to the same one. But anyway, this was for us the proof of principle that really our screening strategy could actually find novel molecular glues. And what about the other 3 molecules? And you will understand in a second why I am presenting the 3 of them at the same time. So same idea here, CRISPR screens to identify the kalium religase, expression proteomics, and in addition we did more stuff here to really pinpoint the mechanism of action. But starting with the target, what we found was that the 3 molecules, structurally different, were inducing the destabilization of the same protein, this cyclin K. And you can appreciate here in these westers that only after a couple of hours, cyclin K is almost completely gone. And this cyclin K is usually in complex with the transcriptional CDKs, CDK12 and 13. So they are required to license processive transcription elongation. So what about the calindrol ligase? So same idea, we did calindrol ligase focused CRISPR screens, and we did find again UB2M, so nice positive control, and we found this partial calindrol ligase complex. So we were very surprised because we did not find any SASTRA receptor. So if you remember, I mentioned that this is usually the component recruited by degraders. So we thought maybe there is an orphan SASTRA receptor that we did not include in this focus library. So we went genome-wide. So we did again resistant screens, and beautifully we found again basically the same. So we started to think that maybe this cyclin K induced degradation was SASTRA receptor independent. And please notice that this will be an unprecedented mechanism of action. So this is the model that we came with. So we thought that our potential molecular glues were inducing proximity between this partial calindrol ligase in the absence of a dedicated SASTRA receptor, and then cyclin K in complex with the CDK. So the CDKs will be acting in a SASTRA receptor-like manner. And we could prove that with our molecules we induce ubiquitination of cyclin K, and this was rescued if we were blocking the CDKs with this non-inhibitor. So it was really CDK dependent. And not only that, so our collaborators in Basel, Nico Thomas' lab, they could use FRET to using recombinant proteins, labeled recombinant proteins, so that only in the presence of our drugs there was an induced proximity between the ligase and cyclin K in complex with CDK12. And to finish, I would like to mention that we were very surprised, and we still are, by the fact that 3 different molecules, chemically diverse molecules, actually converge in the same mechanism of action. And not only that, just very recently, a couple of additional cyclin K molecular glues have been published. So now we are trying to understand how is this possible that all this chemical diversity converges in the same mechanism of action. Also to try to came with more potent probes and initiate preclinical investigations. And with this, simply as a summary, so we are convinced that our comparative chemical profiling using hyponedylated models actually is a strategy that allows, in a rational way, to, for the first time, identify novel molecular glues. And we proved that with the finding of this RBM39 degrader, together with these 3 cyclin K degraders. And with that, I will simply like to say thanks a lot to the Winter Lab, especially to Sophie, Matthias, and Hannah that are here today and that were really key for this project. And simply, I would like to use the opportunity to announce that I will be starting my own lab at the end of the year in Barcelona. So if maybe you are interested in joining a lab in targeted protein degradation, please contact me. And thanks a lot for your attention.
Thank you, Christina. Congratulations again. Thank you for the talk, and good luck for your lab. So lots of success. Wir kommen zum 3. Preisträger in dieser Serie. Da ist jetzt die gesellschaftliche Wirksamkeit auch ein besonderer Aspekt bei diesem Preis. War eine Freude, auch diesen Preisträger anzukündigen, Thomas Gassler. Und ich darf das Mikrofon weitergeben.
Okay, thanks a lot for the nice introduction, and a warm welcome from my side. Good afternoon to this year's online format of the annual ÖGMBT meeting. Before I start the talk, or the main talk today is about the work which we were able to publish in Nature Biotechnology earlier this year, and it was entitled "The Industrial Yeast Pichia pastoris is converted from a heterotroph into an autotroph capable of growth on CO2." And I added the subtitle "From Carbon Dioxide to Vitamins" to highlight one of the application potentials which I will go through during the outlook of my talk. So, on the way to a sustainable economy, we have to face 2 main global issues. So the one issue is the growing world population. Taking different considerations and constraints into account, There are different ways to predict the growth of the world population, but the community is quite certain that we will see a sharp increase until 2050 and that we might even crack the 10 billion barrier by 2050. The other big issue on the way to a sustainable economy is the climate crisis. You all know that, I don't have to go into detail. When we look back over the last 1.5 centuries, we see a sharp increase in temperature each year compared to the average value from the 20th century. So if we now correlate this temperature increase with the atmospheric carbon dioxide levels each year, we see that the rise in carbon dioxide levels correlates with the increase in temperature. And it's just that simple. So, and why is that? So the atmospheric carbon dioxide level is normally held constant in the atmosphere by biological processes on Earth, but also in the ocean. And it keeps increasing because nowadays we still emit more carbon dioxide by the industry that still predominantly relies on burning fossil fuels than we capture by biological systems, and also more than we can capture by industrial sequestration technologies. So that is devastating, and how can we as synthetic biologists and biotechnologists make a difference and contribute to a more sustainable future? So all biotech processes that we now know more or less rely on the use of so-called organic substrates, which are produced by photosynthesis in agricultural farming. And photosynthesis and agricultural farming is of very low efficiency, so we typically see efficiencies below 1%. And this very low efficiency is also the reason why so much land is needed and why biodiversity is threatened if we really want to scale this here up. Then we already see the effects of this scaling up in North and in South America. So what can we do? We can start to use these abundant elements in other ways to turn into electricity. So this would be a sustainable route for that. And this is of very high efficiency. For example, if we take the state-of-the-art photovoltaic conversions that we have today, we reach here over 20% efficiency. We then have renewable electricity, and we can use this to turn CO2 by electrochemistry into what we now call mediator molecules. These mediator molecules can be methanol or, for example, formate or hydrogen. With biotechnology, we can then use either directly these mediator molecules, or we can indirectly use them as energy sources for further CO2 conversion in biotech processes. And this route is exactly what we focused on in this work, is to use methanol as an energy source for CO2 fixation. And we did that by trying, or this was the aim of the study, to turn the heterotrophic yeast Commagatella puffi or Pichia pastoris into a synthetic autotroph that can turn carbon dioxide into biomass fueled by the oxidation of methanol. And I will quickly guide you through how we approach this and how we realize this from an engineering perspective. So native methyllotrophic yeasts can assimilate methanol via formaldehyde in a pathway that we call xylulose monophosphate cycle, and this one is localized in the so-called peroxisome. This C1 assimilation pathway is very similar to what we see in plants and in other autotrophic organisms, and what we call the Kelvin-Benson-Bassam cycle that does the CO2 fixation there. This high similarity between those two pathways has led us to the idea that we can turn the Pichia peroxisome into a synthetic organelle for CO2 fixation. And we did that, so a native or a wild-type Pichia pastoris cell can assimilate and it can oxidize methanol. So the first step in our engineering was to completely block this route and generate a cell that can only oxidize methanol, so constantly generating energy, and it cannot assimilate methanol. And this strain then loses its ability to grow in the presence of methanol and carbon dioxide if you cultivate it, in this case in a bioreactor, in the presence of methanol and carbon dioxide. The next step now was the genetic engineering to integrate a synthetic Kelvin cycle, and we realized that by introducing different genes from different organisms. So in the end, we took genes from plants, from bacteria, but also from yeast, and integrated them into our yeast strain, finally ending up in 3 knockouts and the integration of 8 genes, of which are 6 targeted to the peroxisome. I don't go into detail about the engineering and the genes that we had to integrate, but the key enzyme that was necessary to do the CO2 fixation is the Rubisco enzyme, which is also the most abundant protein on Earth. So if you do that and you hope that this strain then grows under the same conditions that the one before did not, it was very exciting. And I now jump back to the last figure of the paper, because what you see here is very astonishing that this strain really can grow under growth conditions where we can exclusively say everything that we see here in biomass formation comes from CO2 and not from methanol. And it was also very nice to see that it's a very low growth rate, but it's exponentially growing, and it was more or less a proof of concept. So as metabolic engineers, I would say we were not really satisfied with delivering a growth-based proof of our hypothesis. So we went on with a C13 labeling study using C13 glycerol in a batch phase to fully label the engineered biomass. And we saw that on the crude biomass level, let's term it like that, and also in the metabolite pool around the Kelvin cycle with full metabolite labeling around the cycle. We then used C13 carbon dioxide as a carbon source and a tracer molecule, and diluted by growth on this substrate the C13 content from the biomass again. And then we measured again on crude biomass level the C13 content, and we saw that our prediction here with the empty bars matched with the obtained values by mass spectrometry on the crude biomass level, but also on the metabolite level with almost a complete removal after already 1 cell doubling around the Kelvin cycle. Okay, so this was more or less the experiment that really showed that everything that comes, that everything goes into the biomass comes from CO2. But as applied biotechnologists, and I also want to switch the position of us a bit, we wanted to make the system more efficient and more faster. So we applied the technique that we call adaptive laboratory evolution to increase the initial growth rate of our strains. And we did that for several months, and we were able to increase by 2-fold the initial growth rate up to 0.018 for the best performing clones. And we verified the underlying mutations by whole genome sequencing. And in the meanwhile, we also did reverse engineering to trace the effects down to a single point mutation. Okay, I spare you now with more data, and I want to go briefly into the possible application potentials of this technology. So the low efficiency of photosynthesis is mainly due to the low catalytic rate of the Rubisco enzyme. So if you could find improved Rubisco variants, you could possibly increase the yields of crop plants, and with a system that we have here that is relying on yeast, you can easily scale that up, and you could find such improved Rubisco variants using our system. Further, sustainable bioproduction demands for novel feedstocks that do not compete with the food and feed industry. The strains that are reported in this publication are now used to do development studies for the production of chemicals, of bulk chemicals, using CO2 as a carbon source. And we now have proof of concept data for several chemicals using this strategy. The last application potential I want to mention is to use this carbon dioxide derived biomass as a substitute for plant-based biomass for animal feeding, so as a protein source. We now know that our yeast biomass has excellent features as an animal feed protein source. It comes with a high protein content, it has a beneficial amino acid spectrum, and it has a very high vitamin content. So you can easily use that in animal feed. So if you use the electrochemically obtained methanol or formate and you scale it up, you can really reduce the use of agricultural land and make a significant contribution. At the moment, we are working on strains that either use methanol or formate to do the CO2 fixation, and also we are working on strains that co-utilize methanol and carbon dioxide as carbon sources and intended for the production of animal feed protein. Okay, so the great responsibility of our society is more or less defined and combined in the what we call the Sustainability Development Goals, which are defined by the United Nations. With the work in our lab, we identify with several of these goals, and I believe that our work can have a significant contribution to ensuring food security in the future bioeconomy, and that we also can make a contribution to sustainably reduce CO2 in the atmosphere. We further can integrate electrochemically produced methanol and formate into urban and circular economies, and thereby have the chance to generate more self-sustaining and greener communities. And further, by reducing the agricultural land that we have to exploit, we can protect biodiversity with this technology. So with this slide, I want to come to the end of my talk. I want to say thanks to Professor Dieter Matanovic, who became a strong mentor over the years, to my project partners Michael Eggemeyer and Sarah Heinzel, who are here also in the audience, and to all the other co-authors from the paper, especially Matthias Steiger and Michael Sauer, everybody from the lab, especially the ones that are highlighted, and thanks to the society and to the jury for selecting this work and for giving me the opportunity to speak in physical presence this year. It's actually the only physical presence talk I have this year so far. And thanks everybody for listening.
So thank you also for the presentation, und ja, ich glaube, man hat gesehen, der Bogen ist groß, der thematische Bogen ist groß gewesen, von den SDGs bis zum Proteinabbau. Also es ist wirklich eine unglaubliche Fülle an Themen, die wir hier auch abdecken in unserer Gesellschaft. Und ja, ich glaube, es ist mir jetzt gerade geflüstert worden, wie die Frau Bauer vorhin schon gesagt hat, wir müssen mit dem gehörigen Abstand aber doch noch würden wir gerne noch ein Foto machen mit allen Preisträgerinnen und Preisträgern dieses Teils, also die Research Awards, und ich würde gerne bitte auch noch Sie, Herrn Frauscher und Frau Unterer, bitten, mit auf dieses Bild zu kommen, und möchte mich bei der Gelegenheit noch einmal wirklich explizit bedanken für die Unterstützung und Förderung dieser Research Awards. Dankeschön. Gut, dann kommen wir zum zweiten Teil. Wir kommen zur Verleihung unserer PhD Awards, und auch hier haben wir zwei Preisträger. Genauso, wir haben einen Applied Science und einen Basic Science, und wir starten wieder mit der Basic Science, und ich darf bitten, die Preisträgerin, Frau Aglaya Kopp vom IST Austria, uns ihre Forschung vorzustellen. Ich möchte mich auch bedanken in diesem Zusammenhang bei unserem Sponsor THP. Darf sie auch kurz herausbitten, darf sie dann vor allem vielleicht jetzt nicht für den Vortrag, sondern dann für die Preisverleihung. Ja, oder? Ja, jetzt machen wir die Preisverleihung. Ja, genau. Bitte, jetzt bitte ich Sie heraus. Bitte, ja, wir machen die Preisverleihung. Ja, gleich. Die Firma THP unterstützt uns auch als ÖGMBT hier schon seit Jahren, wofür wir uns auch herzlich bedanken. Und wir dürfen jetzt den Preis übergeben.
Das ist der symbolische Scheck, den wir normalerweise überreichen. Ich zeige ihm jetzt nur her. Okay. Was ich aber sehr wohl überreiche, ist ein kleines Goodie-Bag und ich glaube, da ist auch was für einen Kleinen drin.
Vielen Dank für die Unterstützung an die Firma THP, für die langjährige gute Partnerschaft und herzlichen Glückwunsch an die Preisträgerin. Ich glaube, Sie brauchen ein Foto noch alleine, oder? Ja. Ja. Wir sehen uns gleich wieder beim Vortrag, aber wir haben einen zweiten Preisträger, last not least, PhD Award Applied Research, Herr Benjamin Salzer, dem ich auch sehr herzlich gratulieren darf vom St. Anna Krebsforschungsinstitut, St. Anna Kinderkrebsforschung, und herzlichen Glückwunsch auch an Sie. Dieser Preis wird unterstützt von der Firma Polymoon. Ich denke, da ist, glaube ich, heute, soweit ich weiß, kein Vertreter, keine Vertreterin da, aber ich möchte mich in Absenz und vielleicht online sehr herzlich bedanken, auch hier für diese gute Partnerschaft und für die Unterstützung. Vielen Dank. Und herzlichen Glückwunsch. Gut, dann kommen wir schon. Also, Entschuldigung, dann bleibe ich da noch. Dann kommen wir schon zu den beiden Vorträgen ich darf, wenn der technische Umbau über die Bühne gegangen ist, Frau Kopf zu mir bitten für den Vortrag.
Okay, so first of all, thanks a lot for inviting me. I'm very, very happy to be here today. And I will quickly tell you a bit about my research that I did at IST Austria and my main interests. Because I had several interests, but for the sake of time, I will focus on my main project, which was about microtubules controlling cell coherence of migrating cells. And to give you a quick overview of the process that we look at, I make use of a video that was published by Nature Videos some years ago, where you see the cross-section of the skin. So the epidermis is here and the dermis is here. And in there you have a lot of cells. So you have immune cells, dendritic cells, and also T cells. And once there is an inflammation happening, that's indicated here, the dendritic cell gets activated, becomes migratory, is able to locate and enter the closest lymphatic vessel, travel to the next lymph node where it primes cells of the adaptive immune system that in turn leave the lymph node via blood vessels to reach their target site and mediate their effector functions. So in fact, the process that we look at is exactly this. When the dendritic cell is activated and is moving through this interstitial matrix in order to find a lymphatic vessel. So what was depicted here as this relatively loose network is actually very simplified because leukocytes usually reside in very complex environments. So it can be full of other cell bodies like fat cells, or also full of basement membranes and muscle cells or nerve cells. So in fact, inflammation can happen anywhere in our bodies. So that means that leukocytes have to be able to negotiate each and every tissue that they will find on their way. So this means that their mode of migration is referred to as being amoeboid. And this means nothing else that this type of migration is non-destructive, is relatively independent of the substrate, is characterized by having high migration velocities with very frequent and dynamic cell shape changes. So to illustrate that a bit in more detail, we reconstituted this, in that case, the dermal compartment, this collagen-rich compartment, by embedding cells in a collagen network. So you cannot see the individual fibers here, but actually this is a very dense network of collagen fibers, and the cells are embedded here. So when we place the chemoattractant on top, the cell will always try to move towards the highest source of the chemokine. And what you will see when the movie runs is that, first of all, these cells are extremely fast. So the time scale is in minutes. And then second, these cells have extremely weird cell shapes. So they come in so many different flavors, having multiple arms at the same time. But in the end, this doesn't hamper the cell from being very fast. So that means that actually the cell needs to have a very fine-tuned shape maintenance mechanism that on the one hand promotes the existence of a so-called winning protrusion along which the cell will move, and on the other hand promotes the retraction, so the coordinated retraction of all the other protrusions, so arms, that are not needed anymore. So this is why during my PhD research I was addressing the questions, what are the mechanisms that keep these cells in shape? And how does the cytoskeleton contribute to this? And what are the factors that are involved? So what we found was that actually the microtubule cytoskeleton is very important in maintaining the overall cell shape of these migrating cells. Because first of all, these cells, so the amoeboid migrating cells, have the microtubule organizing center located behind the nucleus in most of the cases. And eventually you remember from last year's talk that this promotes the pathfinding of cells, because that way they can use the nucleus, so the biggest and most bulky organelle, to probe for optimal pore sizes along which the cell can move. But in fact what we found that this is not the full story, because actually the microtubule organizing center is a much more dominant spatial selector, because in about 25% of cases the nucleus is found in the losing protrusion. But this is not the case for the microtubule organizing center, because in nearly 100% of the cases, the microtubule organizing center is found in the winning protrusion. So in that protrusion, that will become dominant in the end. And this is indicated here. So when we look at cells moving in this so-called pillar forest, it's a very complex geometry, but it's still so controlled that we can visualize subcellular protein dynamics, in that case of growing microtubule filaments labeled with EB3. So what you can appreciate is that the cell is moving, when the cell is moving, it forms these multiple protrusions on the side that you also can see here in this time projection. But the microtubule organizing center is moving in a remarkably straight line, as if it would already know where it wants to go. So that means the microtubule organizing center fulfills a sort of pathfinding function for the cell. But how can it do that? So for this, we went even a bit more into detail and zoomed in a bit more and looked at processes where the cell is basically making a decision. So the cell is here at this junction point, and before it makes a decision into each of these channels, there are growing microtubule filaments found in both arms. But once the microtubule organizing center switches from here to here, actually this arm will retract soon, so it's going to be the loser protrusion, and this is characterized by having less growing microtubule filaments inside, which is quantified here. So then we were asking, how can microtubule dynamics regulate cell shape changes that locally? And for this, we placed the cells in a so-called path choice assay. So again, it's a complex three-dimensional microchannel where the cell is moving towards this junction point, placing protrusions into all four arms, and then deciding usually for the bigger arms. And we did this in the presence of nocodazole. So nocodazole is depolymerizing microtubules. So we deplete them of microtubules, and we're interested in what happens when the cell has to make a decision here. So what happened is that first of all the protrusion dynamics seemed very weird with these very long protrusions, And in most of the cases, or many cases, the cell actually started to fragment. So it rips apart, which is really bad because soon after the cell will die. And this was interestingly just happening in the presence of multiple dominating, competing protrusions. Because once you put the cell in a straight path, like here, where you don't have any other protrusion than just the winning protrusion in front, then actually nothing happens. It doesn't rupture. The only thing that happens is that it changes direction a bit more frequently. So clearly you need proper microtubule dynamics that mediate this coordinated retraction of the supernumerary protrusions that are not needed anymore for the cell. So shape changes of a moving cell are usually driven by ectomyosin dynamics and not by microtubules. So we were searching for a factor that mediates cell shape changes. So that should be an activator of ectomyosin dynamics, but it also should be regulated by microtubules. And we found that there is one potent ectomyosin activator that's called LFC, or GEFH1, which is the human version. And this is regulated by microtubule binding. So when a microtubule is intact, it can sequester LFC away, it keeps it inactive, no ectomyosin contraction happens. But once microtubules depolymerize, they release LFC activated, and then this activated LFC can trigger ectomyosin dynamics and also cell shape changes. So we were very thankful to receive the LFC deficient mice from the Fischer lab from Magdeburg, and also placed them in this path choice assay. And what we found was that again the protrusion dynamics were weird, and the cells started rupturing. And this was only happening in the presence of multiple competing protrusions. So when the cell just has a single protrusion, sometimes they just place a single protrusion in here and just move on. There's no difference in this junction point passing time. But the moment there's more protrusions present, they cannot be coordinated anymore, so the cell has a problem in deciding which way to go. And this happens in cell fragmentation and cell death ultimately. So together, of course, this was very correlative, but I'm happy to give you more details on that. What we propose is that the microtubule cytoskeleton can actually sort of function as a proprioceptive sense, because it can sense internal cell shapes and locally instruct ectomyosin dynamics to retract certain protrusions in order to sustain overall cellular coherence. And of course, all the studies were done in leukocytes, but there's many other pathological processes that are triggered by aberrantly migrating cells, such as chronic inflammation or metastases. So understanding how these cells basically feel their way through our bodies will eventually help us to reveal new targets for treating autoimmunity and also cancerous diseases. And with this, I want to thank you again for inviting me. As I said, I'm very honored to be here, and I also find it great that you still managed to make it physical. And I want to thank my direct supervisor, Michael, Johanna and Gerhard for giving very valuable input, Efi and Fischer and the Trauner group for providing hands, knowledge and material, and also Jörg for helping with the microfluidics experiments. And I would have one last personal note that actually I'm also very happy to receive this award because it will help to build up the STEM Fatal initiative, which is an initiative that is aiming to promote gender equality in STEM leadership positions. So right now it's not officially launched yet, but you can already sneak a peek at the website or also connect via other channels. So thank you again.
Yeah, thank you both for a great presentation and also for this great initiative, I think, which really deserves attention. I think everybody should have a look and should support this, because I think it's really a great idea, and maybe we have a chance to talk about it afterwards. Thank you. Ja, damit kommen wir zum 5. unserer Preisträger, zum 5. Vortrag von Herrn Benjamin Salzer, den ich herausbitten darf für den Vortrag über Safety and Specificity Engineering of Chimeric Antigen Receptor T-Cells, CAR T-Cells by Exploitation of Avidity. Und
So thanks for this very kind introduction, and also welcome from my side. I would also like to thank the organizers for putting up such an amazing event, and also thanks for giving me the opportunity to present my PhD thesis, Safety and Specificity Engineering of Chimeric Antigen Receptor T cells by exploitation of avidity. So first of all, to get to the same page, a very brief introduction into CAR T cells. So what are these? So that is a new kind of synthetic immune receptors, and in fact, they combine the antigen binding domains of antibodies, so here shown on the top part of the figure, and they are combined with the signaling domains derived from the T cell receptor, short TCR, here shown on the left. And in fact, these molecules enable the MHC independent retargeting of T cells, so a very important class of immune cells, towards tumor cells as a new kind of therapy. And in fact, this first generation of CAR molecules have been in further iterations improved to create second and third generation CARs. But a fact which is very important in CAR engineering and also in the context of my PhD thesis is the fact that CARs, as we know them, are not present in a strictly monomeric form in the plasma membrane of T cells, but rather to some degree in a dimeric form. And that has been shown to be caused by dimerization promoting cysteine residues in the stalk region of CD8 alpha and CD28. And that in turn has been shown to be causative of tonic signaling and exhaustion of CAR T cells, a very deleterious effect for the success of the therapy. So thus we propose to use this effect to do something useful and to create a novel platform of avidity controlled CAR T cells, or short avid CARs. So how would that look like? So just imagine a normal high affinity state-of-the-art CAR, which has a very high affinity and is present to some degree in a dimeric form. You can imagine if this CAR binds to one single antigen, so a monovalent interaction is formed. The lifetime of the interaction is very, very long, and the CAR will be fully activated. If we would use now the same CAR design, but would reduce the single affinities of the antigen binding domains below a certain threshold, we would render a monovalent interaction inactive because the lifetime is very long. But if the same CAR is able to bind to two antigens at a time, we would have on top of affinity a certain contribution of avidity, which is the accumulated strength of multiple affinities, which leads then to the activation of T cells and lysis of the tumor cells. So up until now, it was not possible to control that in a precise manner. Thus, we propose to include three key design principles into CAR engineering to exploit avidity in CAR engineering. So first of all, the usage of low affinity antigen binding domains on top of a monomerized CAR backbone in order to preclude any form of unwanted dimerization. And on top of that, dimerization domain intercellularly driving the dimerization of two CAR molecules. In that case, we would have a CAR system which is dependent on two input signals, first of which being a small molecule dimerizer driving dimerization, as well as the presence of the antigen on the tumor cell. So that is an unswitchable CAR, which is the first application of our platform. But we also can do AND-gated avid CARs. So what would that mean? So just imagine the same scheme as on the previous slides, but now with two different antigen binding domains, targeting two different tumor-associated markers. Again, low affinity on a monomerized CAR backbone. And now, instead of the homodimerization domain, a heterodimerization domain, bringing together two different CAR molecules. Now, in that case, we would have a CAR, which is dependent on three input signals. First of which, again, being the small molecule dimerizer, as well as the antigen combination A and B, and thus also the name AND-gated avid CARs, because these CARs are dependent on antigen A and antigen B. Thus, this form of therapy is way more specific than a normal monospecific one. So in order to test whether this is feasible and possible, we created CARs in two different flavors. First of all, a dimeric CAR with the CD8 alpha present, so we have cysteine bridges, as well as a monomerized CAR backbone where the cysteines have been mutated to serines. As an antigen binding domain, we use the small molecule SSO7D, which binds to human EGFR as our model tumor antigen. And in fact, so you can see the specific lysis of tumor cells expressing our model antigen. You can appreciate that when we use a low affinity antigen binding domain, we were really dependent on CAR dimerization. So in that case, the CAR was dependent on avidity to happen. This effect was not seen when we increased the affinity to higher affinities. So that showed that binding affinities in the high nanomolar range enabled the efficient creation of avid cars. So now we wanted to do that in a controlled and a precise manner, and thus we changed the system a little bit, and we used the same monomerized car, but now with an FKBP12 derived homodimerization domain, which can be very efficiently dimerized by the small molecule AP2187. In order to test whether this is feasible, we turned to an in vivo mouse model using NSG mice, which is currently the golden standard of CAR engineering. And we used the leukemic cell line NAM6 expressing high levels of our tumor antigen. So first of all, here you can see the outgrowth of tumor cells over time, over 11 days. And first of all, we confirmed that CD19 CAR, which is currently used in the clinics, is very, very efficient in tumor control. This was also seen with the control CAR directed against EGFR, which has high affinity, as expected. But when we used the low affinity avid CAR, we lost this tumor control over 11 days. But very importantly, when we induced dimerization at given time points, we were able to restore the tumor control to a level comparable to our two benchmark controls. This was also seen when we looked at the survival of these mice. So we were able to prolong the survival with the on-switched avid CAR to a level which was very comparable to our two benchmark controls. So that showed that conditional dimerization enables the remote control of avid CARs also in vivo. So now the last piece of data. So we also wanted to prove whether end-gated avid CARs are feasible in vivo. For that, again, we had to change the system a little bit. So as already mentioned in the first slides, we used two different antigen binding domains targeting EGFR as well as HER2, so two very prominent tumor-associated antigens. Again, on a monomerized CAR backbone, and then with a constitutive dimerization domain using Lucent zippers driving the dimerization of the two different CAR molecules. So in order to test the specificity of our approach, we turned to a very sophisticated model. So we injected into one NSG mouse 4 different NAM6 cell lines. So in fact, we injected NAM6 cells expressing no antigen, both antigens, as well as the two combinations. And then in the presence of either mock T cells as a negative control, a high affinity CAR as a positive control, as well as our bispecific avid CAR, we looked at the survival of the different tumor cells in target organs. And in fact, after 13 days, we looked at the bone marrow of these mice and then tested for the differential outgrowth of the different NAM6 cell lines. So just to give you an idea how this looked like on day 13, so you can see the presence of all 4 different tumor cells after the treatment with mock T cells, so you can see all 4. When we use the high affinity control CAR, as expected, all cells expressing EGFR, so here on the right-hand side of the plot, were efficiently lysed. But very importantly to see is the fact when we used our bispecific CAR, only the cells expressing both antigens were very efficiently killed, while the single positive controls were spared. Just to give the whole thing numbers, we also looked at the total cells per organ in the bone marrow of these mice. And again, you can see that with the high affinity CAR, here shown in green, all cells expressing EGFR were efficiently killed compared to mock T cells in black. And when we used our bispecific avid CAR, only the cells expressing both antigens were killed, here shown in that part of the plot, while the single positive cells were efficiently spared from tumor lysis. So that showed that end-gated avid CARs enable the specific targeting of an antigen combination in vivo, and to our knowledge, this is by far the most specific approach for end-gated avid CARs. So now let me summarize. So I showed it today that avid CARs integrate inducible as well as logic control functions into CAR T cells by exploitation of avidity. This is based on 2 key design principles, first of which being controlled CAR dimerization as well as low affinity antigen binding. This whole platform can be used in a plethora of different applications. So you can create unswitchable CARs, end-gated CARs, and also end-gated CARs for the recognition of membranous as well as soluble antigens. And last but not least, we also established within my PhD thesis a larval zebrafish platform to test these CAR T cells in vivo. The last 2 things I could unfortunately not include because of time limitations. So with that, I want to thank all the people involved in my PhD thesis and in the studies. and I want to thank the organizers again for having me, and you for your attention.
Ja, danke auch für den letzten Vortrag. Ich glaube, es ist auch hier noch ein gemeinsames Foto geplant für die PhD-Preisträger. Das heißt, ich würde Sie bitten, dass Sie gleich bei uns bleiben und Frau Kopf, dass sie zu uns kommt. Dass wir, ja, bitte mit den Urkunden. Und bitte gerne. Ja, wir haben einen Nachmittag hinter uns gebracht und kommen schon zum Ende unserer Veranstaltung, wo ich Ihnen noch einen ganz kurzen Ausblick geben möchte auf die nächsten beiden Tage. Wie gesagt, das dreitägige Kick-Off-Meeting ist mit dem heutigen Tag eröffnet. Wir haben morgen einen Tag über Cryo-EM in the 21st Century, also im Sinne eines wissenschaftlichen Symposiums, und haben am Nachmittag unseren ersten Life Science Tuesday, den wir mit Spannung und Freude erwarten. Und wir haben übermorgen den dritten Kick-Off Day, da wird es gehen um Aging in the 21st Century. Und am Nachmittag wird es dann um Karriereentwicklung, Karrierechancen im Life Science Bereich gehen. Also ich glaube, ein sehr abwechslungsreiches Programm, und wir hoffen, es ist für Sie alle und auch für alle, die jetzt online dabei sind, etwas Interessantes dabei, und wir würden uns über zahlreiche Teilnahme online wirklich freuen und dürfen Sie einladen, die Gelegenheit zu nützen. Ja, ich möchte abschließend noch wirklich einmal allen unseren Preisträgerinnen und Preisträgern sehr herzlich danken für Ihr Engagement. Herzlich gratuliert haben wir schon, aber auch sehr herzlich danken und wirklich alles Gute wünschen für Ihren weiteren Weg. Ich war echt beeindruckt, also erstens, wie groß die Bandbreite ist, das wussten wir schon in den Einreichungen, aber ich glaube, man sieht auch an den Publikationen, die dahinter stehen, wie tolle Arbeit hier gemacht wird, wie gut auch publiziert wird, und ich glaube, das ist wirklich ein sehr, sehr schönes Zeichen, was wir hier alles im Life Science Bereich in Österreich haben und worauf wir wirklich auch sehr stolz sein können, und herzlichen Glückwunsch an Sie alle nochmal. Ja, damit darf ich allen danken, die dabei waren, die gekommen sind, und ich danke auch allen, die online dabei sind. Von den Kolleginnen und Kollegen, die online dabei sind, müssen wir uns jetzt verabschieden. Wir dürfen hier noch ein bisschen zusammenbleiben mit dem gehörigen Abstand, noch ein paar Augenblicke verbringen. Und ja, ich würde mich freuen, wenn in den nächsten beiden Tagen reichlich Gebrauch gemacht würde von unserem Kick-Off-Meeting, und darf alle einladen, auch in den nächsten Tagen dabei zu bleiben. Dankeschön.